We thought that our goal is to learn the function, right? It's not about learning dynamics is just a means of learning about the function. And then we know that it's a little bit similar to what people have been doing in structure determination. You can learn a lot from sequence using bioinformatics methods, not necessarily based on physical sciences, but simply by some statistics, by some pattern recognition, etc. And it turns out that whether we like it or not, computer scientists have been making a lot of contribution to improving our understanding of the physics, actually, of biological systems, just by simple machine learning algorithms, etc. So we said, why don't we also take advantage of some? So if you want to learn about the function of a protein, the most standard procedure is to see, whether it contains a certain functional motif in the sequence. So why don't we take advantage of sequence information as well to increase our analysis of protein functional properties? So this is the way we see sequence encodes the structure, which encodes the dynamics, which also encodes the function. That's the way I see it there. But it is possible to move from sequence to function as well. Okay? And so sequence encodes structure. Each structure has a unique dynamics. We talked about that already yesterday. And the dynamics is functional. I have shown a few examples. These two are very basic papers which summarize a lot of insights that I'm trying to convey here. Now the question is, since sequences, we know that the sequence encodes the structure based on protein folding concepts. Then if you just complete the circle, can we think that sequences have evolved such that they would encode a structure whose dynamics is functional? So that's the question we raised. So is the sequence optimized, selected to favor functional dynamics, which in turn is actually defined by the structure? Okay, so this was done within the thesis, PhD thesis of a former student, Ying Liu, who made major contributions. So this is the way we approached. Now, suppose you have a query protein. I'm going to take advantage of not only the information in the PDB, but also that in the PFAM database. This is a database of protein families where they give sequence alignments. And these are, you have multiple sequence alignments that have been generated using machine learning algorithms. So why don't we take advantage of that? So we get from the PFAM the multiple sequence alignment. And then we find the corresponding, we have our own sequence here. This is our own sequence. And from the PDB, we have the structure for our query sequence. We can refine the MSA, multiple sequence alignment. And then by examining this multiple sequence alignment, we can make predictions, as I will explain. You can see which particular column is conserved. So you can obtain conservation profiles as a function of residue index. Or you can see which particular pairs of columns co-evolve. They change in a correlated way. So you can obtain what's called a coevolution map, right? And then we would like to compare, whether the conservation profile and mobility, whether they are related in any way, okay? Or coevolution and cross correlations, can we see any relationship? So that was the motivation at this. So and then based on the comparison, we make inferences. Now, intuitively, based on all the discussion we had so far, would you expect a residue that is more mobile, more flexible, moving more, to be more or less conserved evolutionarily. Less conserved. So that's what we're going to observe, actually. So our hypothesis, our expectation is should be reasonable, but sometimes we observe the opposite. But that's what we expected and what we have seen. For coevolution, it's another area. Co-evolving residues are more subtle. They're not conserved. They're not conserved, but they are not totally also randomly changing. They change in a regulated way to maintain some functionality. So we wanted to analyze, where they are, what they do. Of course, we do this analysis for a protein, and then we go back. We did it in a systematic way. It's practically impossible to see the trend by examining a single protein. But now when you have a big set, you start to see features. So but I will show with the help of an example, although the results are obtained, the general results for a big set. Now, for example, for this protein, UDG, first I was expecting to see something to relate to global modes, because global modes are very robust, very meaningful. So this is, and the curves here, the absolute height doesn't matter. I just shifted them. It's just to show the shape. So here is the global mode for UDG, one of the slow modes. I think this global mode would be, I don't remember, but could be mode 1 or 2. And then this is a subset of slow modes. And this is all modes. And the bars here is, they measure the sequence entropy. If the bar is higher, the entropy is higher. What does entropy higher? Entropy is a measure of disorder. So something that's more disordered is more variable, right? So minima here would be the conserved residue, and maxima would be not conserved. And you can already see the mean square fluctuation and the sequence entropy, they do relate to each other, amazingly. This is a good example. Not all proteins are good like that. I chose a good one for you. And then what we like to color code the structure, we color code our structure based on our structural dynamics, the way we do in protein. But we also can color code based on the conservation. Look at the similarity. Amazing, isn't it? So we can learn a lot, actually. If a given residue is a minimum, we don't, usually those minima play a mechanical role, remember? They're not necessarily playing a catalytic chemical role, but a mechanical role. So those mechanically important residues are to be conserved. That's what we are learning also. Sure.
Can you do this with a family, like as a family, that they have a very conserved residues, and can we compare that?
That's what we're doing, exactly. I'll explain more now,
Okay.
because I haven't yet explained. Yeah, and that's the next slide. You are one step the slide ahead.
Oh, no.
So this is the next slide. Now we have, this is what I'm showing here is a multiple sequence alignment. That's one sequence, that's another one. So each row represents a sequence, okay? Now, and these are all family members, homologous sequences. The only way you can align them if they are homologous, okay? So we have, I see that at this particular position, this is all variance, right? This is definitely a conserved site. That site, I+7, is something whose sequence entropy here, based on that, is going to be 0, because it is all, very. Now, then I can see also a few other features. I and I+5. When this is R, this is E. When this is E, this is K. R is positively charged, E is negatively charged. Now, if that mutates and becomes E, probably you want it to be also compensating, undergoing another mutation, and apparently they are interacting. Co-evolving residues are usually, now co-evolution patterns are used for predicting structures because they tell us, oh, it's very likely that these two residues are interacting. So people use that as a constraint for predicting the structure. Okay, so here, and now so I can conclude from here that residues I and I+5 are co-evolving. So if I do this type of coevolution map, this is going to be a strong signal. Okay, it's going to be a peak. So the metric for that, the most standard metric for calculating the entropy is the following. Very simple expression. This is the definition of entropy also in physical sciences and in computer science. So it is at a given position, x here, let me see. Okay, let's say we are at a given position here. We are summing up the probabilities of different types of amino acids. For example, let's take this column. The probability of alanine here is 0. Okay, the probability of glutamine here will be 3/7. Okay, you put the probability of each type of amino acid. You sum over all types of amino acids. And inverse, if you do that type of operation, you obtain the Shannon entropy. Very standard. So you just, in every single column, you have a result for every single column, I. For every column, you have a different entropy value, which you can plot. Okay, very easy. And then for correlated mutations, correlated mutation analysis, it is, the logic is very similar, actually. It is kind of a Shannon-like expression. It's called mutual information, where you have the joint probability of observing a given pair of residues. So you have now a summation over 20 times 20 different combinations. And you need all combinations because you can have A, B, or BA, okay? And the probability, now you are looking at pairs of columns, the probability of observing a given pair, and you divide by the independent probabilities of the individual amino acid types in those columns. So it is like how interdependent are those residues with respect to this independent probability. Okay? So that's the mutual information. So these are the two most widely used, most essential metrics as measure of conservation. The higher the entropy, the lower the conservation. Or the higher the mutual information, the higher the coevolution signal. Okay? These are the most. Now, people, of course, have derived tons of many new methods to improve these things. But this is the basic logic. All the rest is incremental. That's what, that's the essential thing. So that is one improvement, and maybe for the interest of time, I don't want to go into that. But there is a correction here in evaluating the mutual information. I will skip that. But that is also a very broadly used expression for the mutual information, correcting for what's called an average product. So just as an example, and you can, if you are interested, you can see the details. But I think for the timing, if we know those two concepts, it's good for us. All right, one of the intermediate values done in the very beginning of his PhD thesis, Ying did, was to take HIV proteases from a database of sequences maintained by Stanford. And in that database, Stanford University, they have results for patients with the disease that have undergone treatment and no treatment. And then they wanted to, what we wanted to see is what's the difference in the sequence of with and without treatment. So you can, we aligned all the sequences, and we generated this mutual information matrix for the coevolution. You can't see much. Then it is possible to rearrange, reorder things. You can shuffle columns and rows. There are automated methods to do that, such that signals are being clustered. But the residue index is lost here. You just can see some clusters now. So we have been comparing the sequences from untreated, not treated patients and those treated by at least one drug. And then when we ordered, where we can see now better the signals, those treated by a drug had an extra signal here, more co-evolving residues. What are these? What would you expect these to be? Now, these are patients that have undergone some treatment, and now there is a higher coevolution of amino acids in their HIV protease enzyme. What is it? That's what? No, these are not active site ways. This is how your body responds. This is about the resistance to drug. Now, because they have been given drugs, they have mutations for countering the effect of the drugs. This is called drug resistance, or in some cases where you have resistance to multiple drugs, it's multi-drug resistance. Okay? So that is what is happening actually here. So this is this type of coevolution is what has been happening throughout, during the ancestry. This is we call it the phylogenetic cluster simply, but this is the drug resistant. Yes.
So the transformation for both of the matrices are the same. Is the transformation the same? So the left corner.
Yeah. We obtained exactly the same method for two different sets of sequences.
So the lower left is the same.
The way it is reordered.
Correct.
It doesn't need to be. Of course, it doesn't need to be. But then we checked what are the residues here and here. They are practically the same residues. That's one group. No, you need to check, actually, because the ordering doesn't need to be. It is just...
If you didn't check, you can't just say because it's lower left.
No. But now we saw that there is a bunch of residues which are more or less the same here.
Oh.
But now there is a new cluster, and those, when we observe here, wherever they are, they don't show that signal. Okay? This is one way of easily detecting that there is a new cluster, but then you need to check the identity of residues. Definitely, yes.
And in
Yeah.
in that new cluster, do you have similar residues that happen to be in the lower left before?
Here?
Did they migrate somewhere else, or that can't happen? They have to
That can have, yeah, there are changes here too. But one thing I've learned in all bioinformatics analysis, don't pay attention to details. Do you have a rank-ordered list of signals, okay, most co-evolving, less co-evolving, but less strong, etc. Just be very, very conservative. Take the top 1, 2, 5 predictions. Forget all the rest, because then now you have lots of noises. Here, there is no question that there is a feature here. But now, when it comes to comparing these two, I would not, I would refrain from speculating too much. This is the rule, try to do less, but do it more accurately, rather than trying to predict too many things. That's a trap for us, for computational biologists, because we can predict so many things, but many things may not be true. Okay, so we need to be careful. So this is the drug-resistant cluster, and it is located here, actually at the interface. And the phylogenetic cluster, it turns out they are all located here. So it is a very interesting thing to see that they are located at different positions on the structure too. We were very interested in seeing that, the drug is binding somewhere here. This is a dimer, by the way, the protease. It's binding at the interface. And of course, all the drug-resistant clusters also took place at the interface between the two monomers. Now, that was one of the early studies that really motivated us. And then we said, okay, we can learn a lot from these things and then relate to structures and try to understand, to rationalize what's happening. So here, I'll skip that because I'm going to come back to that later on. So then we want to really elaborate more, do more systematic analysis. Are there any key residues? Are they hinges conserved, etc.? These are the questions that I've already asked. Now, coevolution is very, very important because it underlies also adaptability to different substrates. But at the same time, it also confers some specificity. You know, it's not, if it were fully, if that particular residue is fully conserved, it wouldn't adapt to different substrates, right? If it's completely variable, it wouldn't be specific enough. So there is a compromise between the two. I don't know if you can see what I... All right, back to our systematic study. I already explained that. Now, what are the results from that? Okay, and EVOL was based on that study, actually, which you will learn to use today. I don't know why I am having the same thing. Okay. Here you have, now this is the average over 8,000 residues from, I don't know, 200, more than 150 different proteins selected from a database, existing database. And then we have, we calculated for each residue what's the information entropy and what is its effective mobility. Mobility based on the slowest mode or mobility based on all modes. Okay, so we can see very clearly, and now these are we considered grids, different grids of information entropy and evaluated. These points refer to those successive grids. So we can see here this pattern very clearly, especially if the entropy is really high. You have very high mobility. In this regime, it's more or less insensitive. Everything's already fixed. And you can see the pattern actually more strongly in the case of one slow mode as opposed to the average, but you can see the same pattern in both. That's what we were expecting intuitively. You already said that. Okay. Of course, you see these error bars. The error bars get bigger because of the, this is the distribution of proteins that we had. We have very few cases here. This is the number of residues. So that's why we have larger error bars. Now, the question, one question we wanted, and I briefly alluded to that yesterday. Here is a hinge residue. It's not buried. So it's not buried. You would expect something that's exposed to be perhaps not conserved. But it is conserved. These are examples of hinge sites that are evolutionarily conserved to illustrate the case that, the important case that it's not all about surface accessibility. You can have a residue that's surface accessible, but still playing a very important mechanical role, hence conserved. Because not chemical role, mechanical role. So this example shows very clearly that this is lying at the separation between this domain and this domain, if you like, and plays a critical role. And similar features in many other examples. Now, another property, you remember, whenever I'm talking about a coevolution, I'm talking about recognition, substrate recognition, right? Co-evolving sites would be usually really involved in promiscuous interactions in some case, multiple interactions. So when we analyze the coevolution behavior and focus on a given region here where we have many residues that show strong coevolution signals, then we see that those are actually clustered at an interaction site at a region that interacts with substrate. That's one of the examples that I wanted to show. But in general, many co-evolving residues co-evolve because they have to adapt to their interactions in the cell. Okay, here's another example, I think. A very strong signal here when we plot a very strong signal here. Then we also examined the profile, the mobility. mean square fluctuation or the slowest mode, mode 2 in this case. And then we see a very sharp peak actually at this position. These are all the signals to tell you that this residue is involved in recognizing something. It's acting as a flag to recognize, to attract something, and it co-evolves with its near neighbors. And in this case, again, that is that particular region. So this is the protein, the gray and red, and the sign is the substrate. So the region we are observing here is this one, which is quite flexible, actually, to accommodate the substrate binding. So it closes down or opens after substrate binding. So that is another example. So in summary, I'm concluding that part. We have, whenever we have a chemical, I didn't show that, but whenever we have a chemically active site, the mobility will be minimal. So this is what we've learned, in the last 10 years. So it is really important. Mobility will be minimal. It's going to be conserved. What matters at that position is the high fidelity or precision. Catalytic residues, as they have to be really very precisely positioned to undergo their chemical reactivity. Now, the core residues, this is a totally different thing. They usually maintain the stability. They also have minimal residues because they are tightly packed. And now the reason is high stability. It's the opposite, actually. One has to react to, the other has to be very stable, but different reasons for being positioned like that. Hinge sites, again, this is a new group. Minimal mobility. They are like the anchor. This is the hinge, right? It's not, the hinge doesn't move with respect to other sites. And they are conserved. They have rotational flexibility. The things around them move rotationally, but the hinge doesn't translate. Okay? And this is the opposite case. Substrate recognition cases, they have a high mobility, and they show high coevolution propensities, high signals with respect to their near neighbors. More importantly, now, this is something we are in the process of finalizing, but others have also shown that only our lab. This is a very useful metric for finding protein-protein interactions. Now, you know that protein A and B interact. And now you align A and B from different species. And now you observe some pairs that co-evolve. These two residues belonging to A and B co-evolve because A and B make contacts at those regions. So this is now being used as an important method in many studies. I should have given a reference, a review, to find protein-protein interactions. Okay? So that is also about adaptability, as I already mentioned. Okay. So talking about the adaptability, the way we think of modes in the evolution perspective is each mode. I have been saying that modes define the directions of motions, the paths, if you want, paths for conformational changes. So this, and the structure contains those pre-existing paths, which the structure may undertake depending on binding on different functional cases. So we, those concepts we have recently reviewed in a current opinion in structural biology review paper. For those of you who are interested, there's a figure in that review, and I want to walk you through that. That's the structure, the most stable structure that we observe. Now we have multiple substates, 1, 2, 3, 4, 5, 6, for the same protein. That's totally hypothetical, by the way. That's not something, but that's just representative of what we've observed in many examples. So for that protein, that is the, let's say this might be conformation 1, like a Pac-Man. And that could be conformation 2 or 3. So it is a protein composed of 2 domains. And 1, 2, 3 are just different extents of opening, right? And 4 further and 5 further. So if your mode 1, principal axis 1, mode 1, will be this type of opening motion, very likely. Okay. But then there is another state here that's favorable. Oh, so this will be that particular conformation. Now, that's what we predict in the absence of ligands. But this may be stabilized in the presence of different types of ligands. That's the whole point. So that one is stable in the presence of that small, of a mutation that may also stabilize. That's its, let's say, natural ligand, another one, etc. Use different substrates. Now, this is another substrate. And that may perhaps form a dimer. So these are different conformations that the subunit, the protein, may assume under different mutations, complexations, multimerizations. And that is also another example. You can see even a hexameric state like that, where you have this special arrangement. So essentially, the conformational space accessible to the single subunit, unbound subunit, gives you information on what types of structural changes it can undergo under different conditions. So that concludes this part of my talk. And you will see how you can evaluate this type of sequence properties and interpret them in the context of dynamics. So we call it bridging structural dynamics and sequence evolution. I think I already spoke on these things. Oh, as I mentioned in the very beginning, there are many, many different ways of probing sequence, calculating sequence evolution. And we were even confused. We recently did a systematic study comparing different methods. And indeed, you know, you may have many different outcomes, true positive, false positive, true negative, false negative, as you know. And we, in the evolve interface, you have the option of using different two methods. And then what is the utility of having multiple methods? Well, you can consolidate. You can find to see what's the consensus result, right? So I will probably check. So let me, the way we did it, it was to, we took pairs of, we took two proteins which are known not to interact. And then we aligned them, okay, for different species. And then we wanted, we normally, since they are not interacting in the cell, there is no reason for them to co-evolve, right? We don't want those cross terms to be populated in the coevolution matrix. So if these, if we observe ating like that, this is an error. So we want a method that minimizes this type of errors. These are called the false positives. Positive because you see a signal there, but it is false. Okay. So that was one way of you know, gauging whether a method works or not. Secondly, we wanted to see when we examine within a given protein, let's say the protein P, all those signals, we would like most of them to be kind of rationalized based on the structure, to be contact-making residues. Now, if there are others that are not making contacts, that may be equally important. We don't know. But we know for sure that the contact-making residues are, it makes sense, right? So we wanted a method that maximizes this type. We call them true positives, although the others may not be false positives, but we call them true positives when they make contacts, and then we wanted to maximize that. So based on these two criteria, eliminating false positives, maximizing true positives, we tested 9 different methods. And here are the results for those different methods. You can see, so it is, we want more intramolecular signals than intermolecular in the case of non-interacting proteins. And in this case, in cycle, for example, that's what I was trying to say. If you look at the top 0.1% in the rank ordered list, just the top few, they are correct for sure. But then they may become less and less correct as you go down the list. And also this is based on the contact making ones. Psychov and DI, these are the two methods that really give the best result. And I will conclude that part too and move to druggability in the remaining part. So what we've learned is proteins are designed to favor functional changes. There are pre-existing soft modes that facilitate substrate binding. And then the collective mechanics, allosteric dynamics are mediated by those key mechanical sites that are conserved. There is enhanced flexibility at recognition sites, substrate recognition sites. And this issue of specificity, compromise between specificity and adaptability for correlated mutations, that's something I mentioned. Yeah.
So in the plot that you showed, we can clearly figure out if the protein is co-evolving with another system. But what if it's, how do I differentiate between a system with which it's conserved versus a system which is not co-evolving or co-related?
Yeah, it is. So the co-evolving residues, that's a very good question. So the question is how I can distinguish whether a residue is co-evolving or conserved, right?
Yeah, and also with something which is neither co-evolved or
And not co-evolving. Okay, the conserved residues are going to have very high conservation scores, which means very low entropies. Remember, that's the metric. So the conserved residues are going to be, when we plot the sequence entropy, for example, these will be minima. Okay. Co-evolving residues will have intermediate values, actually. They are not fully conserved, they are not fully non-variable, they are somewhere in between. So, but then when you go to coevolution diagrams, in the coevolution diagrams, they will appear as peaks also. You know, when we rank ordered all the pairs, they will be ranked among the top pairs. The conserved residues won't appear there because they don't change. Think about it. Since it's not changing with something else, they won't appear as signals. So you select the co-evolving residues from the top signals in the coevolution maps and conserved residues from the minima in the entropy curves. Does it make sense?
Yeah.
Okay.
Also, sorry.
All right.
What if there is another system which is neither been conserved nor it's co-evolving? So
Yeah.
So how?
Many others.
Okay.
many others. I would say the majority of residues. They will have neutral mutations. So you know, actually, most of the mutations, now as we speak, there are millions that happen. They are neutral. So most of the sites will just undergo neutral mutations. They are not functional. They are not indispensable. Or, yeah. So this is, Julia, what you asked yesterday. That was the thing I was mentioning yesterday. Now we go from small, dialanine, for example, so small peptides to small proteins to bigger proteins or machines. And this is like the number of residues or the size, how many protein subunits you have. So how does complexity scale with the size of the system? That's a question. The thing is, at this scale, you need a lot of chemistry. So this is, you have increasing specificity as you go to smaller dimensions, smaller systems. But as you go to in this direction, you have the dominance of mechanics. So that's the concept I was trying to explain. So we can if we are dealing with really big machines, you can really safely use ANM, GNM. When it comes to small protein peptides, you need to be careful. You may want to use more, specific, detailed force fields. All right, there is this chromosomal dynamics. I'm extremely excited about what is a new development of a student of mine. John, actually, with whom you met. How were the teaching assistants yesterday? They were excellent. Okay. So hopefully it will go very well today. So this is something John did recently. What John did, so these are first co-authors in this paper. This is John. What he did was, you'll be surprised, he applied the GNM to the entire chromatin, human chromosomes. Now there is data on contacts between gene loci on the DNA. So this is called high-C data. So we know which particular regions on the gene, on the DNA, which particular loci that may comprise multiple genes, are making contacts from experiments. So we can build a Kirchhoff matrix for the entire chromatin, for all the chromosomes. And this Kirchhoff matrix can be inverted. You can find the covariance. You can find how the different genes. It's no longer a one-dimensional array, the DNA, right? Now we are thinking of a three-dimensional structure, and more than that, it's four-dimensional. We have its dynamics. So he was able to do chromosomal dynamics using the GNM. So that's just, to me, a fascinating example showing the power of these network models. It was a collaboration. I'm not a genomics person. My collaborator in Kingsford is a genomics person from Carnegie Mellon. I've learned a lot from him. And this was his student. And so this was a collaboration between the two labs to really have a more physical understanding of the entire chromatin dynamics. So using the Gaussian network model. And we were able to explain many observations on genome-wide accessibility, long-range couplings. I will leave it to the end if we have time. But this is really a fascinating study. So we will, I need to go back to our topics. I had prepared that, but maybe we won't have time. So we'll see. Depending on the time, we'll talk on that. But this is to say that you can use those methods in many different disciplines. Druggability, now the new part. Okay, let's start from here. So there is this concept of a druggable genome. We're talking about genome now. You know, the human genome, I think this number keeps decreasing. It is even less than 20,000 genes now. But at least when this review was written, that was the number. Now, the idea is that in your genome, there is not all proteins, your genes are expressed in proteins, not all proteins are druggable. Those genes that express proteins that are druggable amount to a small fraction of all proteins. Okay, this is an important concept, actually, the druggable. And there are those that are at least now, for now, known to be disease-related is another fraction. When we design drugs, we want to design drugs for disease-related proteins that are druggable. So essentially, we have a small target set. And now, one question in computer-aided drug discovery, an important problem is that after even going to clinical trials, they discover that no matter what they do, that particular target is not druggable. So it's important it's an important thing to make an assessment about the druggability of a protein. So that's what motivated us. So these were some concrete numbers when I'm not sure. I think this file is not probably accessible on the web. We'll make sure to make it accessible. So just a small fraction, and only 2% of human proteins right now interact with currently approved drugs. So 10 to 15% are disease modifying, 10 to 15% are druggable, and 5% are both disease modifying and druggable. So that's the statistic. And how the druggable targets are distributed, at least for the time being, what we know, is it's more recent, that one. So if we talk about mammalians, we will have actually, you may notice here, a big chunk. Those of you who are working on membrane proteins, you have good reasons for that. And then there are those in the nucleus, in different organelles, cytoplasm, etc. So that is how the subcellular localization of currently identified 1362 druggable targets. So originally, when Andrew Hopkins wrote this article, he was then working at Pfizer. This was kind of an estimate in 2002, but his estimates turned out to be really quite accurate in a new paper that has been published a couple of years ago. So the strategy usually is, you know, in pharma, they don't do any structure rational things. They do irrational things. They do ligand-based strategies, the most common method. So they have some compounds that they use as a training set. The starting point for all those computations is, now you have some compounds that have shown some activity, right? A few good promising compounds. So this is a good training set to begin with. Now using that, you build a pharmacophore model. So what you do, they use structure actually. They try to see in those compounds that shown some activity, is there a certain chemical feature that's maintained? Is there an aromatic group, electron donor, acceptor, etc.? So they build a three-dimensional pharmacophore model, or it could be even one-dimensional, just features. So then, using this model, they screen existing databases of small molecules. These are called libraries of small molecules. And from there, they try to see whether the pharmacophore model is being represented by a certain member of that database. And then these are called hits. They do those, they do in vitro testing, in vitro, so they get some of them validated, they refine, etc. So this is an iterative procedure. So that's the way it's done. But all this would be, as you can see, in all this effort, they don't take into account the target protein, right? It's all ligand-centric. It's all compound-based. And now we say that we can do better than that if we take into account the structure. So overall, this is you know, like a funnel screening cascade. You gradually decrease the number of test possible candidates until you obtain something, which usually takes a few decades. Now we can our approach was we could do things in a little bit more rational way. Rational way means taking account of the target structure. And if you are aiming at targeting a certain protein, at the end of the day, you would like to understand whether this protein is druggable or not. So that's the question. So this is the HIV proteins that I just mentioned, and this is where the drug binds. Was for those of you in the previous case, I didn't have that. So we would like to understand by structure-based methods docking of small. For example, if we have a structure and we have a target compound, we want to see whether it really fits there, etc. But even before doing that, we want to see whether now we have a target protein. Is this protein druggable? That's the question to begin with. a good thing to examine in the first place is the surface properties. You know the color code here? White will be hydrophobic, red will be negative, and blue positively charged. Can you tell me whether this protein is druggable? The first criterion, you know, the most important thing, it's again physics kind of thing, is shape complementarity. So you need to have some kind of a cavity where you can place something. If it's a flat surface, nothing is going to stick there. You want a shape complement. Can you see anything here? Yeah, you can see that. So this is the drug. You see where it binds? Very. So after you see it, it becomes simple.
Yeah.
So that is how it binds. So the idea is to identify, to see in your protein whether there are the druggable spots. And then, of course, you would like a really good, like a hand and glove type of interaction usually. Not always achieved, but at least. And this is actually a real example, of course, which we were also able to obtain in simulations. Now, what do experimentalists do for druggability? Experimentalists, they just do either X-ray or NMR. When they resolve the structure by X-ray, they try to resolve it in the presence of small organic molecules, and they would try to see where the organic molecules bind. So this would be druggable sites, hotspots. Or with NMR, they also try to examine what are the chemical shifts, where the chemical shifts take place in the presence of the ligands, which would give information on druggable sites. Okay? So we said we're going to do the same thing with computations. We have a protein. We're going to put it in a solution with druggable fragments, druggable small drug-like fragments, small probe molecules, and observe where those probe molecules bind. Okay? That's the same logic. Again, I'm just saying this shape complementarity is very important. You know, kinases have been a very important target. But phosphatases are equally important. One is for phosphorylation, the other is dephosphorylation. Why people don't target phosphatases usually? Well, they are very bad. They have a very flat surface. These are phosphatases. We've been working a lot on them. They are really very hard to identify. But still, you can identify some spots. They are very broadly distributed. It's a hard thing when the surface doesn't present suitable pockets. Okay? Now, what we do is the following in the druggability simulations. I don't know if you can see that. There is a protein here. The white entity is the protein. And the protein now, it's in a simulation box. This is my simulation box. And the simulation, which you will learn in the second part of this workshop. And these little things are the water molecules, which we are just showing like. And then the other things that we show in space filling are our probe molecules. Now, how did we choose the probe molecules? Basically, these are the 4 probe molecules which, at least in the original study, were based on the examination of all FDA-approved drugs and trying to see which functional groups are the most common. So these turn out to be very common, you know, in terms of statistics. So the idea is I'm going to do simulations of my protein in a solution that contains those probe molecules. And I'm going to see where those probe molecules stick. And then maybe there will be multiple isopropanol together with an acetate here sticking somewhere with a very high probability. I'm going to do a statistical analysis. And then based on this, I'm going to build a pharmacophore model. And then based on that, I'm going to screen that against libraries of small compounds to find drugs that represent those pharmacophore models. So that's the plan, right? Okay. So when you simulate, you start the simulations. Originally, you place them just randomly everywhere. But after a while, you would see that, oh, some regions preferably form attract the probes. And then you start to analyze, for example, in this case, MDM2, which is a p53 substrate. There is a region which really likes to bind the probe molecules. Now, this is a schema. These are the probe molecules themselves. In this representation, we're just showing them as small dots, just the center. And if a given region is really very probable, we use instead a big sphere that size scales with the probability. Okay. So this type, this type of analysis worked for me, has proven to identify the druggable sites in many known drug-bound enzymes, proteins. And I'll just show one example here from that was for cytochrome C. And when we were doing cytochrome C, we added, for example, imidazole, because imidazole is known to interact with the heme group, and we want something to bind here. So you do those simulations, and you know, this is like the ensemble of conformations that you have. You can do. The beauty is that you can perform very short runs for that. You don't need long runs, but short multiple runs would be fine. And then from those runs, you are observing which probe molecule has a very high probability of being located where, if it is located anywhere. So that's, for example, something that in this case we saw. Can you see the imidazole here? Against the heme group. This is the heme group. So this imidazole would stick there and stay there during the whole entire course of simulations. So that is certainly, whatever we want to design there, our drug should contain a imidazole group. We knew that. And then there are other also accessory probes, if you like.
Do you have to put the drugs in a particular place to start?
No, no, not at all. We just randomly place them everywhere. And then we start the simulations. So that was showing kind of the simulations. They go anywhere, but now you have already this imidazole that's very stable there, right? It's for sure. Then we do a statistical analysis, you know, all the snapshots. You may not even see them, but this analysis takes account of entropy automatically, because you count the number of incidences if a region is entropically favorable in addition to energy. So now you see that one also sticks to the imidazole. So we start building a reasonable, you know, making some reasonable assessments. It keeps. Yes, because what we're doing here is MD simulation. So the druggability suite of ProD prepares you the files to be submitted, to be fed to NAMD, for example. And also it allows you to analyze the outputs. So it is the pre-processing and post-processing, but in between the simulations are run by MD, by NAMD, for example, or Gromax, the software that you like. Here we like NAMD in this workshop. Okay. Oops. Why is it doing? Oh, I pressed here. I'm sorry? Who is upset?
It's outside.
Okay, all right. So that is, you know, the snapshots from simulations that you can see. And now we using these things, we build a pharmacophore model. Here's what I mean by a pharmacophore model. So you have, most likely you're going to have an acceptor here. You want a ring here, a donor. So these are different features occurring most frequently. So you just define the shape. If you define the shape already, it's a good thing. And then the character, the chemistry at different points. And then you screen that against the libraries. So that is the idea. Libraries, when I say libraries of small compounds, there is something called zinc library, for those of you who are familiar. Or drug bank. We like a drug bank. The drug bank has all FDA-approved drugs. And we like the FDA-approved drugs because they are already proven to be non-toxic. And we have a this is called drug repurposing. This became an important topic these days. The same drug that has been discovered for another purpose can be reused. All right. So that is about, and then we test a small set, etc. Example of a silico hit, again, with our pharmacophore model. That was one of the drugs that we predicted in this example. This is from another, actually, study. But it shows also, the idea that now you screen against this library and you obtain your pharmacophore model, and then you obtain compounds that fit your pharmacophore model. So in that particular study, what happened is that we were extremely proud because we have we screened against 6,000 FDA-approved drugs and 14 million compounds from the zinc library, our pharmacophore model based on these druggability simulations. And then from that, from the first set, we selected 72 drugs. And from the second, we selected 19 compounds. We like better the first FDA-approved drug. And these were tested. Not all of them are testable sometimes, but these were in vitro and also by functional assays. And they, 3 and 4, 3 from the first group and 4 from the second, 3 from the first that are repurposable drugs are listed here. That was also the one I've shown in this example, that, the way it is located. So we came up with 7 novel inhibitors of the peroxidase activity of cytochrome C. In that paper, we also have all the experimental data verifying, which I haven't, some of it is shown here, how those compounds were able to reduce the activity, the top, the most important one, in a dose-dependent way. So then we even did one more study. We said we're going to use imidazole substituted fatty acids to bind, and that was another, this also worked well. I'm going to just skip that. And so the bottom line is that, in general, we can have multiple predictions based on those simulations, which provide good hypotheses to be experimentally tested. You need to experimentally validate, but it's a very good starting point. Now, a little bit more about the methodology here. Remember, the simulations can be very short with different conformers. 10 to 20 nanoseconds of simulations, at most 40 nanoseconds will be good enough. And then you identify the regions, what we call the hotspots. And then we calculate what is the achievable affinity. We do the affinity calculation. So once you generate your MD trajectories, the ProD is going to calculate the affinity for you. Okay, the druggability, druggability. So it does. So we, aggregate the probes, and then we identify the most probable binding sites, the affinities. The way the simulations are carried out is originally you heat up everything. You have a certain, you randomly place them. And then there is an annealing time. You heat everything up, you raise the temperature, you let things relax a little bit. There is an energy minimum equilibration time. And then you have your productive run of 20 nanoseconds. Okay? 20 nanoseconds is a good, reasonable time. And then you start to observe different regions. The whole idea, we are just counting the incidences of the different positions. So you have a series of snapshots, which will be analyzed by DrugGui. DrugGui is the module of DrugGui. Where does the name come from? Drug. And GUI. GUI is a graphical user interface. So that is. So we just count actually, the actual numbers of a probe at a given, lattice site, if you like, at a given grid, versus the expected number based on its composition, the density of that compound. So that is observed. So we take this ratio, and this ratio gives you a free energy. This is a very simplified approximation, actually. But you have all the force fields there already to favor those positions. All right. So here are a few more details. So you could identify actually a very large cloud of a very broad region, and it is, and then we can color code them based on whether it's a cold spot or hot spot. Hot spot means the energy of interaction is very favorable. That's what you can obtain. There is a little bit more about the methodology, but I think all these things will be too technical now. And then essentially you analyze, you identify a compound. You would like it to be a drug size molecule, drug-like size. I will just skip these things. They're showing an example again. This is the real drug. That's what we are finding, the different spots that we obtain. And then when you compare the predicted affinity versus what is experimentally observed. By the way, druggability affinity calculations give you a maximal binding affinity, like a top, most favorable interaction, and it will be usually slightly better than what has been experimentally observed. So this is like an achievable affinity. And we were showing that we did better than other methods. That was, I'll just, so you see, for example, we predict 47 nanomolar. The known affinity is 80 nanomolar. So in our case, but to be even in the same order of magnitude is something important in those computations. So what is beautiful also is nowadays, it is the catalytic site of enzymes is not targeted anymore. You know that? Because a given protein may be involved, a given kinase may be involved in multiple pathways. You don't want to kill the enzyme altogether. You would like to affect a certain pathway, a certain function. So if you want to affect a certain function, rather than targeting the catalytic site and then all of a sudden killing everything, you would like to target the site that is interacting with a substrate along a certain pathway. These are allosteric sites. So the enzymatic site, the catalytic site, is known as the orthosteric, and the others are allosteric sites. So you want to target the allosteric sites, not the active site, a remote. And this method is really very useful in identifying those other sites, multiple sites. So here is an example when we were applying to this protein. There are multiple, sites that are identified. And now, for example, this is the one where it interacts with tubulin, or this is another allosteric site. So we can identify alternative binding regions. That's a major applicability. So these are again all examples from our previous study where we were able to identify those allosteric sites in a series of different proteins. Yes, I'm done practically. Yeah.
Can a user introduce other probes?
Yes, definitely. So one, you can introduce as many as you like. For example, we started with 4 probes. Then we added the imidazole because we knew that it was going to be important. You can add more. The only bottleneck is the force field. So as long as there are force field parameters already available, yes, you can add that as many as you like. And you can you can define your pop set if you like. So I will stop here. And I think I got the feedback yesterday that you like it better to do the application.